Okay, so today we're going to be talking about the cell. Our goals and objectives are to summarize the cell theory, to compare and contrast light and electron microscopes, to compare and contrast prokaryotic and eukaryotic cells, and to describe the structure and function of organelles in animal and plant cells. So there are three statements to the cell theory. All things are composed of cells. The cell is the basic unit of life, and new cells arise from existing cells. So there are two different types of microscopes. The first is light microscopes, which you use the other week in lab, and this will provide you with a magnification up to 1,000 times greater than looking with the naked eye. When you use light microscopes, you will use living organisms. With electron microscopes, they will magnify up to 100,000 times, and there are two different types. You have scanning electron microscopes, and this will provide you with a 3D image. And there are transmission electron microscopes, and this will provide you with a flat looking image, and with this one we can see organelles. Now with both of these microscopes, your specimen will be dead. They actually will be plated with metal and bombarded with electrons, and that will actually provide us with our picture. So here is an image of a mite with a scanning electron microscope, and you can kind of see this 3D image. It looks like it's kind of coming out at you. Alright, so in this slide we have a picture of mitochondria. We can tell this is with our transmission electron microscope. So the mitochondria appear flat, but we are looking at a organelle, so we can see the inner folds in this mitochondria. And then we have a picture of onion cells through a light microscope. So since we're using a light microscope, we know that this is a living organism. And we have here plant cells, so we can see the cell membrane and the cell wall. And you can also see mitochondria here within inside of the cell. So a cell is the smallest living thing that can survive on its own, and there are two different types. You have eukaryotes and you have prokaryotes. You will be responsible for knowing the structure and function of the parts of the cell, and also we're going to talk about the types and functions of intercellular junctions. So in this picture, we have a picture of a eukaryotic cell. So this particular cell measures 10 microns long, and that's equivalent to 10 micrometers, and these are terms that you'll hear later on in microbiology. So in this particular slide, this cell has been stained, and we do that because it better enables us to actually see the cell. So in this slide, you can see the cell membrane, and here you'll see the nucleus. So there are two types of cells. You have prokaryotes, and that's going to be any of your bacteria, and your archaea, which are bacteria-like cells. So these are single-celled organisms, and they don't have membrane-bound organelles, but they do have DNA and ribosomes and cytoplasm. So archaea, they are only different from bacteria in the fact that they live in these extreme environments like boiling sulfur, methane vents around volcanoes, and these environments where other organisms cannot survive. Eukaryotes are comprised of plants and animals. So you are a eukaryote. So eukaryotes will have a cell membrane. Some will have a cell wall, not all. There will be membrane-bound organelles. There'll be a nucleus. And similar to the prokaryotes, you will have DNA, ribosomes, and cytoplasm. So here we have a picture of a typical prokaryotic cell. And you see right here it says there are no organelles. So there are a couple of distinctions that you're only going to see in prokaryotic cells. You may see what's called a pili, and it's this protrusion that goes through the cell capsule and will allow the cell to stick to other things surrounding it. It may also have what's called a flagella, which is this tail that you see here, and the whole purpose of that is to provide the cell with locomotion or movement. This isn't in all bacteria cells, only some. And if you don't see a flagellum, it may have what's called a cilia. It has the same function. It just helps the cell to move around. Now, similar to your eukaryotes, you will have ribosomes because everything needs ribosomes to make protein. So that's going to be something you see in all cells. So as cell size increases, the surface area to volume decreases, and this is why cells should only be a maximum of a certain size. So in larger animals, we have more cells. We don't have larger cells, just more of the cells. This is important, and we're going to actually do a lab later proving this. So important reactions occur inside the cell, like metabolism, and these certain functions are supported by the outside of the cell. So the outside of the cell will provide things to the cell, which they'll actually be taken in and will help with these functions. So inside of our cells, we have a lot of things like receptors, cholesterol. We're going to discuss this more in our next lecture. So it's important for the cell to stay relatively the same size. This expansion can create a higher demand on the cell, and the outside of the cell cannot meet this demand, so ultimately the cell will die. So our eukaryotic cells will have some similarities to the prokaryotic cells, but unlike prokaryotic cells, in the eukaryotic cells membrane, you're going to find proteins and cholesterol embedded in this phospholipid bilayer. That you're not going to see that in prokaryotic cells. So here we're going to talk about the cell membrane more in the next lecture, but we have our polar heads oriented on the outside, because remember, polar heads are going to be more attracted to the water molecules surrounding them, and then those fatty acid tails, they are nonpolar, so they want to face away or get as far away as possible from the water molecules. Now this is actually important because that's what actually helps us establish this membrane that keeps the inside of a cell separate from the outside of a cell. So you're also going to find membrane-bound organelles in our eukaryotic cells, and eukaryotic cells are also bigger than prokaryotic cells. So some similarities that you will see between amongst all cells is that there is a plasma membrane, and it will act as a selective barrier. All cells will have cytoplasm. You're also going to hear it called cytosol. It's the same thing. All cells will have chromosomes, and chromosomes are what carry genes in the form of DNA, which we're going to talk about more later on as well. And then all cells have ribosomes. Remember, again, we are protein-making machines, and we need ribosomes to do this. All right, so here we have a picture of an animal cell, and you'll see that we have a cell membrane and we have some cell membrane bound organelles within the cell. A couple of things that help distinct animal cells from plant cells. You see here that we have a centrosome, and a centrosome is just a pair of two centrioles. We also have lysosomes, which is specific to animal cells. Animal cells also have the ability to have a flagellum. You're not going to see it in most animal cells, but you will see it in some. A good example of this is a sperm cell. I think everyone's probably seen an artistic rendering of a sperm cell, so you can think of that little tail that's coming off and helps provide it with movement. All right, so in this picture we have a plant cell, and one thing you'll notice is that the plant cell is more square in its shape, as to where your animal cells are more the round. So one reason for this is that plant cells have a cell wall, and you can see that cell wall surrounding the cell membrane. So that's one thing that will help you tell the difference between a plant cell and an animal cell. Plant cells also have a central vacuole, and this holds a lot of their organic compounds, as well as a lot of water, and the central vacuole actually takes up most of the space in this cell. Plant cells will also have chloroplasts, which will help them in the process of photosynthesis and making their own food. And plant cells also have what's called plasmodesmata, which is their particular kind of an intracellular junction, and it's how the plant cells communicate with each other. So the nucleus is where you will find the chromatin, and chromatin is just simply DNA combined with proteins. So we know that DNA will not leave the nucleus, so anything that needs to happen with the DNA is going to occur inside of the nucleus. Now the nucleolus is a condensed region of chromatin where ribosomes are synthesized, and again, ribosomes are used to make proteins, and again, we need proteins for everything. So ribosomes are made of RNA and proteins, and they're formed in the nucleolus. And these ribosomes help carry out protein synthesis. So we have the endoplasmic reticulum, and we have two different types. The first type is smooth ER, and smooth ER helps to synthesize lipids, metabolize carbohydrates. It stores calcium, as well as detoxifying poisons. So the organ that's responsible for detoxification is the liver. So in liver cells, you'll find a higher concentration of smooth ER than you would compared to cells located elsewhere in the body. So our rough ER is similar to the smooth ER. The only difference is that there are membrane-bound ribosomes. You can actually see some membrane-bound ribosomes right here on that endoplasmic reticulum. So rough ER, again, has those bound ribosomes, and it helps in the production of proteins and membranes. The Golgi apparatus can be thought of as the post office of the cell. Its main function is the modifying, sorting, and packaging of proteins for secretion. It also plays a role in transporting lipids around the cell. The lysosome can be thought of as the stomach of the cell. It digests the macromolecules that you get from eating, and so there are two ways of doing this. There's phagocytosis, which is called cell eating, and there's pinocytosis, which is cell drinking. And you can remember the two by remembering that Pinot is a type of wine, which of course you drink. So you can help to remember pinocytosis is cell drinking versus phagocytosis, which is cell eating. So our lysosomes are also referred to as suicide cells because they can go through autophagy, and that enables them to do what's called apoptosis, which, go ahead and get familiar with the term, is programmed cell death. So basically, the lysosomes will commit suicide, and whenever a cell is either mutated or if a cell is infected, the body can, or these lysosomes can enable the cell to commit apoptosis, again, programmed cell death. So vacuoles will actually fuse with these lysosomes, and that is to help feed the cell. So the cell needs things that will come from outside of the cell, which you can see that happening here. This would be a great example of phagocytosis. So these lysosomes will bind to this food vacuole, and then will actually have enzymes that break down these food particles. So as we talked about this before, plant cells have a central vacuole, and this takes up most of the room in the plant cell. This is where their water is stored, some other organic molecules that they need, but this is mainly water, and we'll talk about this a little later on. But this is actually what helps with turgor pressure, which is what enables plants to have the shape they do, to stand up. So different cells have different ways of making energy. Our two main forms are the mitochondria, which will be found in eukaryotic cells. Actually, will be found in eukaryotic cells and prokaryotic cells. And then plant cells will have chloroplasts, which again they use to perform photosynthesis, and that's how they get their nutrition. So the mitochondria is enclosed by two membranes. It has a smooth outer layer, and then it has an inner membrane that's folded into cristae, and you can see the cristae right here, and you can also see them over here in this slide. So this cristae is where we have cellular respiration, and cellular respiration, don't actually think of breathing. Cellular respiration is how when we break down food and we break the bonds of those macromolecules that we've taken in, this is where it comes into play. The energy of those bonds will be used to make more ATP. So chloroplasts can be found in plants and algae. It can also be found in some single-celled organisms, but it's usually going to be an indication of a plant cell. The Chloroplast contains chlorophyll, and it's the site of photosynthesis. So we also have perioxysomes, and the function of perioxysomes is oxidation. So they actually produce hydrogen peroxide, which is then converted to water. So we have a cytoskeleton, and that's comprised of microtubules, intermediate tubules, and microfilaments. So a cytoskeleton is a network of fibers that's found in the cytoplasm, and it provides support, motility, and regulation. So without a cytoskeleton, there would be no shape to a cell. It would basically be flat. So microtubules support and grow out of the centrosome. And a centrosome is just a pair of centrioles. So microtubules actually make up flagella and cilia, which you should remember me talking about with prokaryotes. These types of cells will use these structures to help them move. So both flagella and cilia are made up of microtubules, and they're in a 9+2 structure, meaning that there are 2 centrioles that are surrounded by 9 centrioles. So microfilaments help with bearing tension, and they resist pulling. These are often found in microvilli. And a common place you'll find microvilli is in the lining of your intestines. There are these finger-like projections, and this is probably some kind of molecule that's going to be taken in for digestion. Another type of or another use for these microfilaments are by amoeboids. So amoeboids actually move around through, or they use what's called a pseudopodia to move. So pseudo means false, podia means foot. So they will actually grow this false foot, and they'll actually pull themselves forward. Microfilaments also play an important role in cytoplasmic streaming. So if you were to make a slide of a plant, you would actually see this occurring. So in this particular picture, we have a picture of a plant cell. We know that because we see our central vacuole. We also see chloroplast. Now, if you were to pretend that this were on a slide, if you left it in the light long enough, now not so long that it kills the cell, but enough for the chloroplast to actually get enough energy, you would see the chloroplast moving around the cell, and that's how it actually delivers its nutrients. So the plant cell wall is made of cellulose, fibers, polysaccharides, and proteins. Remember, we cannot actually digest cellulose, but it is the fiber that we need in our diet that we get from plants and vegetables. Remember that cellulose, because we can't digest it, it actually helps in excretion because it takes some of the workload off of our intestines. Also, there are some cells that have multiple layers of cell walls. So animal cells have no walls. They actually have an extracellular matrix that's made up of glycoproteins and macromolecules, and this provides support. It allows the cells to adhere to whatever it needs to. It also helps provide movement and regulation. So let's talk about intracellular junctions. Now, in plant cells, we have what's called a plasmodesmata, and that's just these little junctions where the cells are bound together, and this provides communication between cell to cell. Animal cells communicate through 3 different types of intracellular junctions. The first being tight junctions, and this is where one cell is affixed to another so tightly that they can actually talk. This also prevents the leaking of extracellular fluid coming through. This is important in the microvilli of our large intestines. We don't want water or nutrients leaking into other cells. Then we have desmosomes, and this is where cells are anchored to one another, and you're going to find these in keratin and the hair and your nails. Then we have gap junctions, and these are communicating junctions, and they have special proteins, and they surround a pore. So this allows ion sugars, amino acids, anything to go from one cell to another. All right, so let's do a quick recap. We talked about the cell theory. We compared and contrasted light and electron microscopes. We compared and contrasted prokaryotic and eukaryotic cells, and we talked about the structure and function of organelles in animal and plant cells.